Hello and welcome to another episode of Momento Malum today. Throughout the history of agriculture, there has been a gradual refinement and corresponding erosion of the crop gene pool. In modern times, the lineage of modern cultivars can be traced back to artificially inbred genetic lines, which are then crossbred to make F1 hybrids. In the case of naturally cross-pollinating species, as well as genetically pure selections from primordial land races and the derivatives of hybrids between such selections. Corn is an example of the former. This video will cover the pedigree and genetic history of the inbred lines utilized in the creation of new inbred lines and by proxy F1 hybrids of corn. Let's get started. In 2001, Dr. Romero-Severson created a paper utilizing the pedigree data and SSR genetic marker technology within multiple corn genetic lineages. The goal of this paper is to determine the relationship between the genetic inbred lines of different corn subspecies such as dent, flint, popping corn, and sweet corn. To achieve this goal, the researchers obtained 57 elite inbred lines from North America and Europe of both university and commercial origins. These lineages can be grouped into multiple clusters. These groups are the Iodent group, Northern Flints, the sweet corn, the European Flints, and popcorn populations. The results of the study revealed that the European Flint and sweet corn inbred lines are derived from the Northern Flint lines of the northeastern United States. The inbred lines of popcorn seem to be distinct from the other genetic lines, but are closer to each other than many other lineages. The inbred lines of the B73 group were founded on the inbred line B73. This line was derived from the cultivar group called the Iowa Stiff Stalk Synthetic, and it's through B73 that all other inbred lines of the B73 group are made. Because all B73 genetic lines were made by crossbreeding B73 of other lineages, and then artificially inbreeding the resultant hybrids. The Iodent lineage all have 29% or more of their pedigree derived from the genetic lines known as Iodent, Long-Ear, and Minnesota 13. With minor traces of other lines mixed in to their lineage. The pedigree of these lines will appear in the next slide. Later on, another paper was written on the same topic in 2011. This was a research project made by the AAFC and Dr. L.M. Reid. This project analyzed the genetic diversity of 129 Canadian corn inbred lines, using both pedigree data and SSR genetic marker technology. Of the 129 corn inbred lines, 119 of these were bred by the AAFC itself. The rest are foundation stock and historic inbreds that are used to breed the 119 genetic lines. Just using the pedigree data, of the 129 genetic lines, 94 can be placed into 8 distinct groups. The 8 groups are derived from the following genetic sources: the Iowa Stiff Stalk Synthetic, European Flint corn lines, the Lancaster Open Pollinated cultivar, the Minnesota 13 inbred line, the open pollinated line known as Early Butler, the Iodent Synthetic, the inbred line known as Pioneer 3990, and the Pioneer inbred line known as 3994. Group 1 is predominantly derived from the Iowa Stiff Stalk Synthetic. Group 2 is derived mostly from the European Flint lines. Group 3 is derived from the Lancaster Open Pollinated cultivar. Group 4, Minnesota 13. Group 5 Early Butler, Group 6 Iodent, Group 7 Pioneer 3990, Group 8 Pioneer, and Group 8 was derived mostly from Pioneer 3994. The researchers could not categorize the remaining 5 lines using pedigree data. However, through the genetic data, a more complete picture could be created. Using this data, the researchers could divide the inbred lines into 10 genetically distinct groups. Group A was derived from the Iowa Stiff Stock Synthetic and Early Butler. Group B was a mix of all genetic founders. Group C was derived from a combination of Lancaster and Pioneer 3990. Group D was derived from the mingling of the stiff stock synthetic and an unknown group. Group E is derived from the mingling of the European Flint lines with at least one unknown genetic group. Group F was derived from Minnesota 13, Early Butler, and Pioneer 3994, as well as at least one unknown genetic population. Group G was derived from the Lancaster open pollinated cultivar. Group H is of unknown origin but can be placed into 2 separate subgroups. Group I was derived from all groups except for Minnesota 13 and Pioneer 3990. And Group J is of unknown origin. Within these pedigree groups, the average genetic similarity between all inbred lines was 66%, and Group 2 and Group 6 had the highest level of genetic similarity. And that about covers everything. Thank you for watching. If you want to donate to my channel, the links are below. If you want to buy my book, you can also find that link in the description box below.